Hello friends, all are welcome to my channel Get Easy Medical Micro. In my previous lecture, I discussed the gram-positive bacterial cell wall. Now, today I will discuss the gram-negative bacterial cell wall. In case of gram-negative bacterial cell wall, we seen it is more complicated structure than gram-positive bacteria, and we also seen in case of gram-positive bacteria, it is 80 to 90% of the cell wall is covered by the peptidoglycan layer. But in case of gram-negative bacterial cell wall, not only present peptidoglycan layer, it also present, but it is very less in amount. How much? It is 10 to 20% is total covered by the peptidoglycan layer, and other few chemicals component are there. They also covered up around the bacterial cell wall. Okay, and it is the main component is peptidoglycan, which is present and which is the thickened of the gram-negative bacterial cell is about 10 to 20 nanometer. Okay, now if we see the gram-negative bacterial cell wall, it is divided into the total cell wall is divided into 4 different parts, and it also subdivided in another part, mainly 4 part. Okay, one is first is peptidoglycan layer. Number 2, lipoprotein. Number 3, outer membrane. Number 4, lipopolysaccharide. These 4 major different layer is present outside the cell wall of gram-negative bacteria. Okay, clear? How it is formed? Now we have to see. Now it is formed. Let's draw the picture. It will be very easy for you. So we should remember same thing, the cytoplasmic membrane always be present, and outer part of the cytoplasmic membrane also present the cell wall. Okay, so here same like that, gram positive bacteria, this one is the cytoplasmic membrane. So this one is cytoplasmic membrane. We draw the picture of protein lipid, and also here also called the cytoplasmic membrane. Okay, so this one is cytoplasmic membrane. So we draw the picture. So okay, this one is we already called cytoplasmic membrane. Okay, cytoplasmic membrane, this one. So We already said four different layer is present, and it also subdivided into two or three part, but major is these four. Okay, these four. Now let's draw where this layer is present. So first one is peptidoglycan layer. It should be reminded that in case of gram-negative bacterial cell wall, peptidoglycan layer is not closely attached with the outer portion of the plasma membrane or cytoplasmic membrane. So what is this? Peptidoglycan layer is not closely attached to the plasma membrane. Not closely attached to the plasma membrane. Suppose this one is peptidoglycan layer. Okay. It is not closely attached. In case of gram-positive bacteria, we see peptidoglycan layer is more thick and it is closely attached to the cytoplasmic membrane. But in case of gram-negative bacteria, it not closely attached. It is present on floating condition over the periplasmic space. So it is present over the layer like this. Over the layer, it is present on floating condition over the layer. Okay, over the layer. This is called the periplasmic space. Plasmic space. Okay. So what is the function? What is the importance of periplasmic space? In this periplasmic space, so many degradable enzymes. Degradable means some molecule can enter inside this. It can easily break it easily, or chop it. So if any chemical compound, any another material, any solute molecule which can passes through the bacterial cell wall, if it enter inside this periplasmic cell, it will easily degrade. So this is the main function of periplasmic space. It has, in this space, have so many degradable enzymes. Clear? So peptidoglycan layer is present on floating condition. It is not directly attached to the cytoplasmic membrane. Okay. So this one is first layer of gram-negative bacterial cell. One is peptidoglycan, and you also see this one is very thin. It is very thin than the gram-positive bacteria. And in case of gram-positive bacteria, it all are covered by the peptidoglycan layer. But here is only 10 to 20%. Another one, number 2 is lipoprotein. This is the lipoprotein. If you its molecule is compound with the lipid and protein molecule. Lipoprotein. This is the second layer of the gram-negative bacterial cell wall. This lipoprotein is closely attached to the peptidoglycan layer. Inner portion of the peptidoglycan layer. Just it. Inner portion of the peptidoglycan layer. What is the function? Its main function to provide the stability. of the total gram-negative bacterial cell wall. Okay. Here, it is present with closely attached with the inside of the peptidoglycan layer. Okay. Number third, which layer is present? Outer membrane. Outer membrane. Okay. Outer membrane. So, outer membrane, clear? Outer membrane outer membrane. Outer membrane. Okay, which one? This one is outer membrane. This, let finish the draw. This total portion is outer membrane, and this small portion is called the phospholipid layer. This one is outer membrane. What is the function? It also provides the stability of the gram-negative bacterial cell wall. In that layer present so many porins protein. This one is porins protein. This one is porins protein. P-O-R-I. Porins protein. And it is also called the transmembrane protein. So what is the function? So many chemical molecule which can get easily enter inside the gram-negative bacterial cell wall, it can easily pass through this porins protein. Okay, clear? When we treated the antibiotics, okay, so that time it can help to pass inside the gram-negative bacterial cell wall. So porins protein must be present, and it will allow to molecule enter inside. So this is the porins protein which generally present in the outer membrane. That is the function. And phospholipid, and they also function to provide the stability. Okay. Now this is the third layer, outer membrane, and fourth layer is lipopolysaccharide. This portion is called the lipopolysaccharide. This portion is called the lipopolysaccharide. It is closely attached with the outer membrane, very closely attached. Okay. This lipopolysaccharide mainly subdivided into three parts. Subdivided into three parts. One is lipid A. Okay. Number two, core polysaccharide. Another three is O polysaccharide. D. So this lipopolysaccharide is subdivided into three parts. One is lipid A, core polysaccharide, and O polysaccharide. Okay. Now we discuss these three. Lipid A generally associated with the lipopolysaccharide layer. Okay. Lipopolysaccharide layer. Like this. What is the function of lipopolysaccharide layer? It is act as a endotoxin. When bacteria will die, it can produce endotoxin. Okay, when bacteria will die, that's the effect of pyrogenicity, tissue necrosis, and immunoadjuvant like this. Okay, so this is act as a endotoxin, lipid A. And next is core polysaccharide. Core polysaccharide is, it has not generally has not function. It closely associated with a lipid A, and it also produced by the some sugar molecule, and it also provide the strength of the lipid A. Okay. Next three is O polysaccharide. O polysaccharide is present outside the lipopolysaccharide layer. Okay, outside the lipopolysaccharide layer. What is the function? If it is also called O antigen. O polysaccharide is also called O antigen. Why? If this O polysaccharide is present in gram-negative bacterial cell wall, the bacterial colony will be smooth in nature. Smooth. If it is absent, then bacterial colony will be dry in nature. Okay, so it is one of the identifying features also in gram-negative bacteria. Okay, and sometimes it also disappear, O polysaccharide and also O antigen. It also sometimes disappear when repeated subculture. At the time of repeated subculture, it will disappear. Okay, that is the major function of the O polysaccharide. So this one is total structure of gram-negative bacterial cell wall. Here now is seen that peptidoglycan layer. This is present inside the all other out of bacterial cell wall. It is present inside. Okay, so it will be very helpful when I will discuss the gram staining procedure. How gram staining procedure is happening and why that it will take to different color. So that picture will be very helpful to understand easily. Okay, clear? So this much is enough. Thank you.